Sure. Hi, everybody. My name is Ethan. It's September 22nd, 2021. Drexel AI's first GBM for the fall term for week one. I'd like to welcome everybody to Drexel AI. We are an academic interest group that focuses on teaching, mentoring, and researching in the realm of artificial intelligence and machine learning. I'm happy to see all the new faces. So the way this usually works is we treat our weekly meetings like project meetings. So we usually review progress for a project that we've been working on. As of right now, I don't think we have a project in mind yet. Savek has proposed a few, which we might go down that route because I think they're particularly interesting, but we can talk about that later. First of all, I'd like to give a bit of background about the club and myself. The club was started in 2019 by Yit, who is now a graduated Drexel alum. He was studying computer science and he did some research in machine learning. I think his master's was on something called adversarial attacks, which was particularly interesting. Since then, we had the e-board elections last year, and I was elected president, Alicia, vice president, Savek, and as treasurer. I think Adam is the event coordinator, and he's not here right now, but you might see his face now and again. So yeah, like I said, my name is Ethan, and I am a third-year biomedical engineering student studying like the intersection of machine learning, software engineering, and bioinformatics. I find that intersection particularly interesting. I have a background in a little bit of AI, mostly genomics, and now I've been working at a medical device company called Moberg Analytics. Riddick works with me there now, and we focus on creating software tools for treating and managing traumatic brain injury patients in clinical trials, and it's particularly interesting. So I'll pass it off to Alicia. Would you like to give an introduction?
Sure. Hi, everyone. I'm Alicia. I'm the vice president of Drexel AI. I'm a third-year computer engineering major, minoring in math and neuroscience. I was a part of this club since fall 2019, which was my freshman year. My experience with AI, I've done neuromorphic computing research with one of the Drexel professors here, Dr. Anoop Das. I'm currently doing bioinformatics with Ethan. We're in the similar lab. I've done computational neuroscience. I have like a stronger background in neuroscience. And recently I'm trying to get into like sparse coding as well, which is like another realm. And yeah, that's all I have. So I'll pass it on to Satvik, who's our treasurer.
Hello, everyone. I am Satvik. I'm the treasurer of Drexel AI. I am with Drexel AI since last term in the summers. And I mostly work with AI, and specifically I work in the computer vision, and I apply computer vision with the medical imaging modalities such as MRI scans and CT scans and X-ray scans for more of the diagnostic predictions and outcomes. And I have previously worked at the Georgetown University with the same diagnostic prediction making. And as of right now, I'm working at the College of Medicine with Professor Osborne on neurocomputing. And I'm, again, looking forward to working as a sparse coding as Alicia and into neuromorphic computing. So hopefully we'll see some of that in the coming terms.
Sure, you can pass it on to anybody, just popcorn it out.
Okay, I'll pass it to Abby.
Hi, I'm Abby. I'm a computer science major, and this is my first year at Drexel. My interest in AI is I'm fairly certain I plan to do my concentration in like AI and machine learning for computer science. So I thought I'd join because I'm very interested in AI, even though I may not have much to contribute at the moment.
No, welcome. We invite, older, younger, less experienced, more experienced, everybody. Call on somebody else. Abby?
Oh, okay. I don't know anyone here.
It's okay. Just like say a name.
All right. There's a Stefan. I don't know if that's how you say it. Oh.
Yeah, Stefan's fine. Don't worry about it. Hello, my name's Stefan. I'm a third-year computer science major. I'm very interested in AI algorithms and learning new ones and trying to understand how and why they work. So I also love math and all that stuff. And I've been doing some research lately with Professor Anoop Das on spiking neural networks, and that was also very exciting. Alright, then who's next? Why not Riddick, if that's how you say it. I'm sorry.
That's how you say it. So I'm a senior at Drexel University right now. I'm a computer science major and entrepreneurship minor. This will hopefully be my last year if I don't slack on my credits. Right now I'm on my third co-op at Moberg Analytics with Ethan, and I'm interested in working in the DevOps and intersection of DevOps and machine learning. So basically DevOps for machine learning. And I'm also working on my senior design project right now, which involves identifying eosinophil cells using CNNs. I'm just starting right now, but we'll see how that goes.
What kind of images are you using?
Sorry?
What kind of images are you using?
Just images taken during research. I haven't even looked at the images yet,
Okay.
so just starting out.
That's cool. That's cool.
Yeah.
Sure. Welcome.
Next, I would like to call Shivang. How about you?
Sure. Hey guys, I'm Shivang. I'm a freshman and I'm planning on double majoring in finance and math. But I'm also really considering switching it to finance and comp sci because I like comp sci a lot. My background is, so in high school, I worked as a web developer. So that was like my main thing, but I'm trying to transition over into AI more. So this past summer, I did a little bit of self-studying, but I'm still like a very like beginner stage, like in that AI and all that. So I'm hoping to learn a lot more and get to know everyone a little bit better. I'll popcorn it over to Pernille.
Hi, I'm Pernille. I'm a sophomore computer science student. I'm pretty new to AI myself. I actually joined the club over the summer, but I didn't get to interact much. So I'm excited to learn more. Why don't we pass it to Nayef? Is that how you pronounce your name?
Yes. Hi, guys. My name is Nayef. I am a master's student. Hopefully this is my last year majoring in AI and machine learning program. I hope that I can participate more in the club this term. I have like 6 years work experience in software development, DevOps, and project management. I did several projects in deep learning and machine learning. The last one was about stock market prediction using deep learning algorithms, LTSM, and yeah, that's it.
Sure. Welcome. Glad to have you back.
Thank you.
Do you wanna pop it off to somebody?
Yeah. Alicia.
Oh, I already introduced myself.
I'm sorry. All right. Kevin.
Hi, I'm a fourth year computer science student. I'm also doing my senior project with Riddick, funny enough. I'm a AI and computer vision dual concentration with a minor in mathematics. I've done a bunch of ML and DL projects. I recently took 615, even though I'm an undergraduate student, which is deep learning. Brilliant class, totally suggested for everyone here. As for the senior project, I remember, Ethan, I recall you asked a question about what kind of images we were doing. They're digitized slides. So basically you're looking at slides on a microscope and you digitize what you're seeing in that microscopic like zoom. So these images are like 10 gigs each, which is massive, even though it's only for like 2 centimeters worth of like actual data. So one of the major things that I was looking forward to was, finding good ways to pre-process those images so that you could actually feed them into like a neural network. And on top of that, perform image segmentation on top of it to actually determine, the cell count in nasal tissue, since eosinophil cells are like nasal tissue cells. And apart from that, I've done projects with, detecting COVID when looking at X-ray images. I've done various projects, such and such. And yeah, hopefully I get to contribute more this term towards the club.
Yeah, of course. Welcome. You calling somebody?
Right. Looks like pretty much everyone. How about Srija? I don't know if I said that correctly.
Hi, my name is Srija. I am a sophomore right now, majoring in computer science. I don't really have a lot of experience when it comes to AI, but I'm really interested in it, specifically in machine learning. So yeah, I joined Drexel AI just so that I could learn more about it and interact more with everyone here. So yeah.
Cool, welcome. You can call on somebody.
Oh, okay. Dubem, I hope I'm saying your name right, but yeah.
Yeah, you said it right. So my name is Dubem. I'm a second year software engineering major, and I joined this club because I just find AI very interesting, although I don't know much about it.
Welcome. And then can you call on the last person? That's a.
Is it Yashi Jane?
Yes. Hello. Hello. Am I audible?
We can hear you.
Hi, I'm Yashasvi Jain. You guys can call me Yash. I'm a computer science major and a neuroscience minor. I'm really interested in brain-machine interfaces, and I was going through a lot of projects over the summer. That's where I got interested in AI, and I don't know much about it though. So I'm here to learn and contribute in any capacity I can.
Cool. Welcome. Awesome. We won't do introductions every meeting, I promise. Just the first one, because I think it's important to just get familiarized with the faces, because people are going to be in and out depending on their schedules throughout the term. So 2 things. The first thing is that we'll be attending the involvement fair, not tomorrow, but on the 30th. I'll be there, and if anybody else wants to swing by to hang out, be my guest. I printed out the papers that we worked on last year, a QR code to just submit your emails, and then Savek and I both have some things we want to show. He just sent me a zip file. I don't know what's in it, but I have this program called PyMol, which is effectively for viewing 3D protein structures. And I kind of want to have that up so I can explain the one project we did last year with protein folding. So the second thing is an introduction to how we operate. At least how we operated last year is we have a GitHub, and from this GitHub, I can add you guys. So first step, if you don't have a GitHub, you should please make one, and then DM me your names on GitHub so I can add you as a contributor. I think these are all, I think I have to add you as a contributor for you to work on the repositories. So just add me, or sorry, just send me your name on Slack and I can add you. Sure. So, right, to get you an idea of what we've done before in the past, I can pull up some of our papers. I want to get in Yeah, so fall term, this was a pretty interesting paper what we did. We worked on an optimization algorithm to find the best number of hidden units in a single layer neural network. We did that by applying binary search on a very well-structured problem. So you can imagine if you have a basic setup like this, where you have, n hidden units in the first hidden layer, you want to optimize this according to some metric. So if you're looking at accuracy, you want the number of hidden units in the hidden layer that optimizes accuracy. We did that by applying binary search on a very well-structured problem. So we assumed a distribution of accuracy with respect to the number of hidden units, and we implemented a searching algorithm that effectively does what binary search does, but on a distribution like this. The next one we did in fall term that year is we looked at traversal algorithms. So a limitation of this one is we assumed that the number of hidden layers was 1. In this one, we assumed we didn't make that assumption. So we were looking at different traversal algorithms to find the best number of hidden units and number of hidden layers for a more generalized case. And before I go on, I want to mention that this paper was accepted last year to the International Joint Conference of Neural Networks and presented at a conference held through Harvard. So a lot of the papers that we write and a lot of the projects we work on, there's upwards of like 7-ish authors here, 6 authors, 7 authors, and there's no limitation to the number of authors we put on a paper either. And this one was published at 2 venues through conference proceedings. In the winter term last year, we did a project on stock market prediction. This one didn't do as well. We were trying to use the convolution operation as an objective function. An objective function is just something you try to optimize during like backpropagation or through like an optimization algorithm. The idea is that if you have a stock market pattern, right? This might be something called like a consolidation breakout, where you're consolidating for 10 time steps, whether that be days, minutes, seconds, milliseconds, depending on your granularity. You want to be able to predict that this breakout is going to happen in the future. And what we hypothesize is that, well, if you can find evidence of this occurring in the past, can you somehow match the pattern of this to a dictionary of these empirically defined consolidation breakouts? So for those who don't know, the convolution integral, it basically measures the overlap of 2 signals. I won't go into it now, but basically the function results in a maximum when the 2 signals are exactly overlapping. And we were using that as a function that we would use as optimization to find this matching pattern. I think our implementation was a little off in the results. It followed that our results didn't do too well. Still a project nonetheless. In the spring term, was this the spring term or was this summer?
The protein one was spring, I think.
What did we do?
Summer was
Evo
the time.
What did we do in
Time-
summer?
The time.
Oh,
Evo
yeah. Okay,
Time
sorry.
series.
Sorry. Okay, so yeah, spring, we worked on the protein folding problem. So I don't know if you've seen in the news, it was a big hot topic. The people who made AlphaGo made this program called AlphaGo is an AI model for solving and beating world champions, grandmasters in the game Go, which is it's a board game. Not I don't know the rules, but apparently it's harder than chess. They developed AlphaFold, which is for solving the protein folding problem. So in bioinformatics, the protein folding problem is defined as, the input is an amino acid sequence. So different amino acids and just a string, no chemical structure whatsoever. And using that amino acid sequence, using the string, can you predict the three-dimensional protein structure? So how it looks in three-dimensional space, when those amino acids fold in each other in like in a soluble solution. So we explored this using a generative adversarial network, which is a two-player game. You have a discriminator and a generator. The generator generates images, and the discriminator tries to discriminate those images, those same images. And the idea is that they're trying to beat each other. So the discriminator is trying to get so good that it can tell between these two images, and the generator is trying to get so good that the discriminator can't do that. And because of this zero-sum two-player game, they're supposed to both optimize each other. So this is what an optimization might look like. You might have the objective function of the generator increase and then decrease, and then you might have the discriminator loss decrease and then increase, or something similar to that. So the idea to this is that we'd give protein structure, an amino acid sequence, something like that, and we'd try to get a generator to generate that, but then also get a discriminator to discriminate between a real protein and a fake protein. And I think our definition, the paper was lacking because our definition of what was real and what wasn't exactly clear. I think we aimed for real proteins as what we call native structures, and native structures are just the optimally folded protein structure, and then a decoy structure is just everything else. That's flawed because, as you can imagine, there are so many more decoy structures than there are native structures. So it's kind of biased in that way. And then this summer, we worked on a project that I was really hoping we'd complete, but I've just been swamped with work. It was on time series forecasting. So it's the ability of, given features, in the past, can you predict the future? So univariate time series prediction. So a univariate single step time prediction. So if you can imagine, like, you have maybe, I'm going to draw red. You have some time series, it stops here, and you're trying to predict a single point right here. This has been shown to work relatively well in literature. What doesn't work so well is when you use these features to predict like a multi-step problem, only given univariate input right here. So the idea with this was to combine sparse coding, which is an unsupervised model that finds features that describe the data, common features that describe the data, and forms a dictionary of those elements. And our idea was to do predictions using an LSTM, and then also find a dictionary that describes those same elements, and iteratively use that dictionary to find the weights that best predict the output. And we did this in a generative manner, where we try to segregate the data into different generations, and we wanted the weights to basically improve as you went from generation to generation. When we got the results, there was negligible improvement, if any, but that might just have been because I was implementing it and I may have messed up. So with all of these projects, there's definitely room for improvement. Most of the time when you implement an algorithm, you have to make a couple assumptions. So you're kind of limited on, on how well it works. when you ever implement a model, you can always be short-sighted for, biases or cases in which the model just won't work. So with these projects, I mean, these are topics in AI, relevant topics. I mean, their applications are, I haven't gone much into them, but they are pretty far-reaching. The idea with the time series prediction one, that one's a little more generalized. If you can predict time series, it doesn't matter the units. You have opened up a range of possibilities because, most periodic signals, especially biologically related, behave more or less the same way. It's the same general trend. When you think of an EKG, right? It's the same trend, but it's just shifted up and down, and the spacing's a little off, depending on. Then this is how you measure heart rate. So if you can predict this, you can apply it to intracranial pressure waveform. Which is the pressure beneath the skull. So it operates in a similar periodic fashion, where there's these three peaks, but they just move up and down, and they might space out a little bit more. So that was the main motivation for that. And I could go on for a lot of I could go on and on for about the applications for the projects, but I think with it being week one, we've gone over introductions, we've gone over last year's work. Does anybody have any questions about the projects or the general gist of what we want to do? If
There's
not,
a question in the chat. Sorry.
Is the dict supposed to represent a genetic encoding? Kevin, do you mean the dictionary for the sparse representation?
Right, for the one with the evolutionary sparse coding project that you have mentioned.
Yeah. So sparse coding is a very, very cool, very, very hot topic right now. The atom, sorry, the dictionary is composed of these things called atoms, which are just dictionary elements, and each atom is supposed to represent a common feature of the data itself. So it's not tagged to any label. It's just supposed to represent, a patch of the image. If you can imagine that we're looking at images of cats, like you just zoom in on a cat's face, a patch might just be the cat's nose or the edges along its face, depending on like how zoomed in you're looking or how big the patches are. So the dict represents a collection of explainable units, and Savek is tagging a good source from Stanford. It's a really cool algorithm. I hope that makes sense. And the main motivation for sparse coding is you don't want to have something that you could train those same images on like a deep neural network, but there's two problems. One problem is, what if your images aren't labeled, right? What if you're dealing with a supervised problem? So what if you're dealing with an unsupervised problem? So the difference between supervised and unsupervised, you don't have associated labels. So you don't know if a picture of a cat is a cat, or if a picture of a dog is a dog. The second problem is that the weights of the deep neural network, so the associated contribution of each feature, it's not too interpretable. I haven't done much research on that, but that's the general trend. You can't explain why a given layer. So like, let's say you have a neural network with 6 layers, you can't explain the weights of the connections off of the 3rd layer. It's just, I just really don't, I doubt that you'd be able to do that. So the sparse coding is supposed to solve both those problems, or provide an alternative method to working around those problems. It's a cool algorithm. Good question. If you ever have any questions about like the techniques I mentioned, just like let me know, because I've gotten pretty comfortable with like tossing them around, but I know most people aren't. And I the best way to understand something is to explain it. That's kind of why I like teaching a lot, because it brings me a lot of joy. So yeah, just ask. There are no more questions. we can start talking about what we might do this term. Safek, you had a good project idea. It has to do with your background, and I think because I haven't done image processing before, and there's a lot of things you can do with image processing that I'd like to learn about. I know pretty surface level about image processing besides just throwing it into something called a CNN, a convolutional neural network. Yeah, do you want to talk about it?
Yeah, so I had the idea of doing something with any sort of medical imaging, and maybe we could go, as I was seeing this year, like I went to a few conferences and I saw the trend moving towards pathological images more than the medical imaging. So maybe we could look up for a data set, because I was looking through the cancer archive and there were like a few fresh data sets for pathology. So maybe we could work on that, because that would be a state of the art. We could use the off-the-shelf model unit, which is specifically for biomedical images classification. And it would be relatively an easy project, not that tough, because we're using an off-shelf model and just implementing on a data set. And it's very beginner friendly, I think. And even Alicia, I guess, have experience with that. So maybe we could consider doing that.
Cool. What kind of images are we talking about?
Wait, let me pull up.
Sure. I'm not familiar with the data set or the model.
Okay.
Which are two very important things.
Yeah.
Yeah. So
Okay.
is there any novelty? Would there be any novelty in that?
Yeah. So
Which part?
the data set itself is very novel because it's not yet classified yet. So that is the novel part. And then applying units is also would be novel because it's a current hot trend.
Okay.
So yeah, I would just,
Sure.
Or we could also do a cancer MRI or X-rays. That is also okay.
Let's pick one image modality because image
Yeah.
processing, you can get lost in the details, I'm sure, if you try to do
Mhm.
multiple, because it's very important we focus on one kind of imaging, because MRI and CT are very different. Just
Yeah.
by like CT is a cross section of the brain, or
Mhm.
one of them. And MRI just looks like the other way, I think. So
Yeah.
it's different, and I want to focus on one thing.
Okay.
The reason I ask about novelty is usually when we try to publish papers, we want to incorporate something that hasn't been done before. It gives it boosts your ego to do something people haven't done, and it's better for if you want to take the project to conferences.
So if you want to do something really novel, that would be if you take a data set, medical data set, and try to make a synthetic data set using GANs.
Yeah, that would be novel.
That would be pretty novel.
So is the data set you're talking about, do you have the link to it?
I'm just opening it. My net is pretty slow right now.
Okay. If you get it, can you send in the chat? Because I'd like people to see it.
Sure.
Yeah.
I read a paper, I think, and I remember there was a model, like a UNet GAN model. It's like I
Yeah.
think the discriminator of the GAN is like a UNet based one, or is it a generator? I don't know, because like there's a certain GAN that like uses like a similar architecture to a UNet, but I don't remember if it was the generator or the discriminator part of it.
But the one you sent to me for the breast cancer one?
No, it was a separate one, I think.
Okay.
I want to pull up a picture so that people aren't lost in what we're talking about.
I'm just 1 second trying to open the data set.
So when I say GAN, it's called a generative adversarial network. Generative because you have this generator. Adversarial because I think the reason it's adversarial is because you're throwing in fake images. And the network because you have these two models. And it's a game between the generator, which generates a face from random noise, which is being compared to real faces. So this discriminator has to improve such that it can, discriminate between the real and the fake faces. And as the generator gets better and better, the discriminator will get worse and worse. So the idea is that their optimization should counteract each other, and they should both simultaneously become better. I've never used UNet. Is that just a deep neural network for medical images?
It's like
Yeah.
it's similar to a convolutional neural network. It's just like slight changes, basically.
Yeah, so often when we talk about deep neural networks, the architecture is pretty important. And when I say architecture, all it means is just the number of hidden units at each layer and the number of hidden layers, and then something called an activation function, which is how the numbers change from each layer. And there's different activation functions, and then convolution has its own weird way of behaving. But this is a generally, an architecture for UNet, which I guess was optimized to do well. And I wouldn't be able to explain this architecture. I'm really kind of lost, but.
This architecture was actually specifically built for 3D images, so something like MRI
Okay.
scans.
Yeah.
Okay. That's
And what
why.
it does is it does like a cascading effect. So basically it breaks down the 3D images into a bunch of blocks.
Mhm.
And from those blocks or patches or whatever you want to do, it builds itself back up, and then it returns an output from there.
Interesting. That's cool.
Yeah, and they like specifically designed it mainly for like biomedical images.
Cool. So,
Because, sorry, one of the limitations of doing like neural networks with medical image data sets is that medical images in general are really big. Like
Yeah.
you're talking about like stuff that's like either not even just like discount the fact that it's 3D models. Usually you'll have like a volume, which is like a bunch of images stacked on top of each other, because you're not really operating in like RGB space anymore. You
Yeah.
need to have more color information to get everything. So medical images usually have their own implementations for breaking it down. And that was actually going to lead me into another question I asked. If we're going to do an image data set for medical purposes, how are we going to deal with the fact that most data sets require a lot of images, all of which are really big?
yeah. So I had an idea. I could, I can't do much about the big, I can't do much about the sample size. I know that, deep neural networks require a large sample size. I can't do much about it. Unless you were to segment an image into smaller images, and you could classify or somehow predict some metric about patches of an image, so you'd be able to increase your sample size that way. That might be an option. Another option is to deal with the feature, just the sheer amount of features you have. You don't need all of those features from a given image. Like right here, it's really hard to see, but you can see how a lot of this right here is like dark space. Point in the upper left-hand corner. Most of it's dark space, but there's this little tiny sliver that is important right here. And then like right here is also important, right here is important. So I'm kind of wondering if, you could use something to select those features. There's segmentation, which has been used, but that's not exactly automatable. But you could also use sparse coding to find out which features are most important, which would build a dictionary of relevant explainable features, which you could feed into a deep neural network. And I don't know of anything that's used a sparse coding model and then a neural network in tandem like that to have the sparse coding algorithm learn the features and then use those features to predict that label.
Would sparse coding be applicable for multi-dimensional images like this?
I guess so, because
yeah.
I read a few papers on medical imaging, and I guess Professor Kim does work with ultrasound images, right?
Yeah, he does.
Yeah. So I think it's pretty much applicable.
And also like there was I remember like with Dr. Kim, there was during one of the lab meetings, someone presented a sparse coding for like a medical image for a GAN model too.
Sorry. We have a question. Do we always involve real images into the data set from the generator? Or is it only in problems where we were trying to tell real pictures from fake? So the whole point of the generator, the GAN, is to simultaneously create a model which can generate images and create a discriminator which can discriminate those two images. So when you implement this model, you kind of get the best of both worlds, because you can generate those images and you can also discriminate between those two. So you'd want both. It's just the nature of the architecture for this specific problem. Yeah, this site does not exist. It's cool. This face does not exist. It's yeah, so this is the results of a generator generating fake images of people, right? And I guess the idea is that you refresh it and all of these people don't exist, but they look like people. Like, I guess that's the idea. And it's a really cool application. Like, this guy doesn't exist. This guy doesn't exist. These are all fake. I knew that existed. I just didn't know where to find it. Thanks. So going back to the medical images.
Ethan, I also gave one more website, like one more data set. It's for radiogenomics. So it's basically, I don't know if you could do this, but it is medical imaging features grouped with the genomic features. And it is for lung cancer cells.
Okay.
That's cool.
It looks cool.
Are these CT scans or like?
Yeah,
MRIs?
these are CT scans.
Okay.
And what genomics do you have? What other assays or the sequencing?
I'm just going through that. These are RNA sequences.
RNA-seq, okay.
Yeah.
So the way RNA-seq works, it's to study gene expression. RNA-seq.
It's at the bottom if you see.
Oh, is there sample data?
Yeah.
No.
You could search.
Oh, it has clinical data too.
Yeah.
It's pretty fun.
That's what I'm saying. It's really fun.
Yeah. So RNA-seq basically measures the expression of a given RNA strand, and it's spiky because it tends to have high expression in certain places according to where the genes line up. So if you have a gene right here, it would transcribe maybe this gene. You'd expect the expression to be high, something like that.
And there's also a paper that uses for a CT, I think it was a CT images, one of those optimization algorithms. I think it was artificial bee colony for like feature selections. Yeah, I think it was CT, but I'll try to find it and link it, because I also try to use it for my own. Like, I did a brain tumor project with the UNet, and I tried to use that same type of method. So
yeah. So, all right, I like this data set a lot, and I want to use it. So we have a data set, right? We have a data set. We just need to define a problem, like something we're going to predict. And it can be exploratory for a little bit, because usually in the past we've struggled to find data that's as nice as this is. I mean, you have 210 patients, and you have much more than just images. You have, clinical data, and you have genomics, which is amazing. Like, wow, that's called multimodal data.
Multimodal data.
Yeah.
Yes.
And my company works with multimodal data, so this makes me very happy. So, I mean, thinking about the type of data we have, right? I'm gonna just use my annotator. I always draw. I like drawing. We have CT. We have PET, CT, annotations of tumors, systematic too, controlled vocabulary, that's good. Segmentation maps, of those CT scans, quantitative values from the PET scans. So these are like metrics of the PET scans, can be used as features. Paired with gene mutation, RNA sequencing data from the tumor site. Clinical data, survival outcomes. Okay. So does anybody have any ideas?
Um,
It's okay if like none of you guys know what we're talking about. Like I know
Ask questions.
people said like
Yeah.
they have like not a lot of experience, but don't get intimidated by what we're talking about. So,
Yeah. Thanks, Alicia.
Quick
Yeah.
practicality question.
Sure.
How are we going to actually access this data considering the size of the actual, like the entirety of the data set? Like it's like 100 gigs. So where would we store something like this? And how would we even read this into memory? Like, would we read it in by batches?
Yeah, you'd have to do batching. I had a similar problem with protein structures, very large. You can actually just use like a generator function to read in to batch it like one sample level at a time. It works out okay. It just takes a lot longer to train. So Alicia and I have access to Alicia, do you have access to the HPC at CHOP? Did you get?
no, not yet.
Oh, okay. So I'm associated with the Children's Hospital and I can use their high performance computing machine, which is a supercomputer. Oh, I don't know. Okay, sorry. It's not a supercomputer. It's a cluster of really powerful computers. I won't misuse supercomputer like that. That to be a disfavor to the supercomputers of the world. It's a really powerful cluster of computers, and I've done a lot of models on there. I did my protein structure model on there last summer. So I'd imagine that I would just download the data there, and we could run the models there. It's more about right now formulating like a problem statement. So we don't not have the computational bandwidth to take on a data set this big. And you don't even have to use all of it, right? I mean, it's 100 gigs, but that's including all the modalities. We might only just use, one of the images. I mean, one question I have is, so how much of the genomics data can actually, well, okay, I want to step back. So there's okay, if you're in space, right, there's this saying that you can't see any man-made structures from outer space. And the reason that is the case is because you're so far, and what we make on Earth is just so minuscule compared to the grand scheme of things. So, I mean, I have this question, like, does that hold? I mean, it's not exactly the same. It's kind of it's like an analogy. It is, if you were looking at a medical image, can you see can you see the genomic phenotype from a medical image, and how much, and if you can, you probably can, how much of the genomics can explain the variance in the image data set? So I'd be particularly interested in looking at how well, like, the RNA sequencing data and the gene mutation data can explain observations observed in like CT scans.
Like what comes to my mind when I see like imaging, clinical data, survival outcomes, which I find very useful, is if you could work on precision medicine, because precision medicine is basically these three terms. And if you could work on basically treatment decisions, like approach to treatment decision, like if this person, we are looking at one scan and then come upon various features, we say whether this person needs radio, chemo, or just nothing.
Yeah, I mean, that would, I'm sure that would require expert knowledge, and that's not included in the data set. Depending on the data.
Do we not do that with your sparse encoding or something? Feed it into like a random forest or decision tree model?
What was the first part of that?
Like, could we not use either the sparse encoding or your RNA sequencing, like whatever's provided, either, process that somehow and then throw that into either a random forest or a decision tree model and figure it out from there.
I mean, yeah, but what would be the associated feature you're trying to predict? Would it be something from the imaging data? Would it be
I'm assuming we would have to see. I'm not sure if this is like labeled with what exactly the segments are. Because if it's labeled, we could probably figure it out. But if not, I don't think. Yeah, it definitely wouldn't work out that way.
Yeah, Suffolk. Yeah, good point
Ethan?
though. Yes.
You can hear me, right?
Yes.
Oh, good. Okay. Oh, I have another question.
Mm-hmm.
So, were you asking if we had any ideas of things that we could work on before I left?
Yeah, we're talking about this data set.
Oh, okay. Yeah, okay, just making sure because I wasn't sure what you were asking before.
Sorry.
Yeah.
Yeah. We're basically looking for a path to go down this term for a project. And this was created, June? It's created 2 months ago.
Exactly.
Do you know of what has been done on this data set?
It's just went like public like few days back.
Yeah.
Yeah. It was like ongoing, like since the last I checked, last month.
Yeah. Check the citations. Oh, never mind. That's policy.
No.
We could look at a more general approach, like what actually has been done with AI in radiogenomics, and then maybe apply some useful ideas from there.
Yeah, I've actually shared an article, and I'm sharing one more, which is actually a review. So, oh, no. I sent the same one again. I'm so sorry.
That's a good point, Kevin. Sorry, I usually like to jump into things.
Kevin, Ethan, this one would be actually better. The one I sent now, NCBI one.
Yeah, I was just saying because like it doesn't make sense. Like I've never worked with radiogenomic data before, so I don't know what projects have been done on it already. Yeah. So maybe looking at others, we can grab some inspiration off of that.
Yeah, no, that's true. This, as you can tell, it's like insanely new. So yeah, this is exactly what we need to do. And funny enough, I'm working on a grant right now that would do exactly this, radiogenomics. So I need to at least learn.
It's a perfect project for you, Ethan, then.
Yeah.
Yeah.
Are you looking to hire people?
It's through my lab at the Children's Hospital, and that's the association between me and Alicia. Yeah, we're short staffed, and we're looking for motivated people who can spend a considerable amount of time with us. But you're Suffolk, you're busy.
I might be free.
Yeah. So cool. Kevin, good suggestion. We're coming up on 7:00. I'm gonna read these papers over the next week. I'll probably read them tomorrow. And I'll probably read them tomorrow. I suggest that other people, read them, try to get through them. If you're interested in it, go for it. Yeah. Usually if I read something that's AI related that I don't know what it's about, there's this YouTuber called StatQuest. He's absolutely amazing.
I watched videos for my 615 class.
Yeah, he explains everything. He explains everything very simply. Josh Starmer. Like so many stuff, like PCA, neural network, clearly explained, linear regression, p-values, so many things. So I would go there first and just Google around, but I'll read these. If you don't have access to this, I don't know.
I might send it on the Slack. I can send it on Slack.
Can you send the papers on the Slack? I don't know if it's because I'm like logged into a VPN right now or something, but I just want everyone to have access to this stuff.
I'll send it. And Ethan, can you send a link in the chat if people are not on Slack? Like an invite or something?
Oh, yeah, sorry. I was assuming that everyone's involved in Slack here.
Oh yeah, and if you guys have like some suggestions, not like of like either projects or like research areas you want to like dive
learn more
into,
about.
just let us know. If you don't want to do like
Yeah.
just medical stuff.
Yeah. Savek and I are kind of hotheads for medical stuff though. But a lot of the stuff applies to other areas because eventually, you know, people who are coming new into AI, you'll realize that there are a bunch of different algorithms to use, and not every algorithm works in the same set of problems. So after a while, you start building up a tool belt. So, you know, principal component analysis might work in some cases. Random forest might work in other cases. Neural network might be good, but then it's kind of black box. So then you need something that can pick out the important features like sparse coding. Like it's just how I learned is I did projects that focused on individual algorithms at a time, and I kind of saw where they did well and where they didn't do well, and I try to just build a tool belt like that. So that'd be my advice. Cool. So I'll read these and I'll start thinking about what we can do. I mean, the simplest thing as a target variable might just be to predict clinical outcome. I don't know how measurable that is. Sorry, I don't know how reliable that is. I wish that they had matched control samples in this data set. That's one thing that's kind of, it's a shortcoming to this, because usually when you want to predict something, it's good to see what that like a negative control is. So when something isn't present, so if you have a disease, right, if you have cancer, you want images of not cancer to compare as a baseline.
Yeah, right.
Yeah.
We don't even have to be tied to this data set. We could probably find some other medical data sets that also fit the purpose of what you're looking for.
Yes, this is true.
Yes.
This data set's amazing.
Yeah.
So
also, if like
yep.
anybody is feeling uncomfortable with any kind of like AI thing, machine learning thing, you could reach out to me on Slack. I have plenty of material to send. You could reach out to me with questions and stuff. I'm happy to help.
Yeah,
especially
well.
medical imaging and stuff.
Yeah. So for all those interested, there is a channel on Slack called Medical Imaging.
Oh, weird.
I'll say join. Can you paste them in Medical Imaging?
Yeah, I'll do that. Yeah, I'll do that.
Oh no. So please join that if interested. I won't make a GitHub repo this week because I don't think we have a clear idea of what exactly we're going to look at. I kind of want everyone to look at those papers, you know, read them, look over them, ask us questions in Slack. Usually, so my lab is very active on Slack, or I won't say that. My company is very active on Slack. We always talk to each other, and I'd like people in Drexel AI to be as communicative. Just, you know, it's very easy to reach out and to me or in a channel about a question, and you know, we have 185 people on here. I know they're not all active, but I'm sure someone will have free time to, you know, to join in the conversation, and generally people are friendly, so that's good.
Also, when is the involvement fair? Is it 23rd or is it 30th?
30th.
30th, okay.
Cool. Awesome. Yeah, Kevin, good
Quick
suggestions,
question.
by the way. Yep.
Appreciate it. How do you feel about NLP on radiology reports?
For biomarkers?
Not necessarily biomarkers, just, you know, I guess it could be for biomarkers, you know, identifying what, like, let's say there's an image and it comes with an attached radiology report, and you're trying to figure out, you know, what is the status of this person's lungs, or for example, looking at the radiology report.
Kevin, have you looked at medical annotations before?
I'm not a medical guy at all.
Okay.
I'm just coming up with hypotheses.
Yeah. So one interesting thing is that, you know, NLP is called natural language processing. It's for analyzing text. It could be in books, it could be speech. So when you actually look at medical annotations, it's a lot more difficult because it's not natural. If that makes sense, like the language you're analyzing isn't natural. So I don't know whether conventional NLP would, or the NLP that works well on natural language would work well for annotations, because annotations tend to be a lot of abbreviations, a lot of incomplete sentences, a lot of, you know, nurses scribbling things down. It's a much more difficult problem. My team at Moberg, the company I'm at, had a data analyst from Massachusetts General Hospital do NLP, quote unquote NLP, on medical annotations, not restrained to, you know, scans like you're suggesting, Kevin. And it was a pretty difficult task, but it would be worth explaining if that data set was available. Ideally, you'd want all of these modalities paired for the same patient, because then you could get more of a holistic view. You want as many modalities for a patient as you can. And that's kind of what my company Moberg is working on for traumatic brain injury. But that would be a interesting point. But the sooner we pick a data set, the quicker we'll be able to get going with the project, and, you know, get people to, and maybe when we dive into a project, into a specific problem, people will get a better idea about the whole, you know, scheme of machine learning and all that. Yes.
Actually, at the Amy Lab at the Stanford, they did a similar project with Professor Christopher Manning. I have just sent a link to it. If you're interested, you could look at that. It was a pretty complicated project, though.
Yeah, NLP. I think that's a misnomer, but cool.
I barely could understand that, but yeah.
You could barely understand it. Oh no.
Yeah.
Cool. Awesome.
Yeah.
Okay. So homework, just look at those papers if you're interested, and I'll start thinking about, you know, possible projects, and we can brainstorm, of course. If you haven't sent me your GitHub username as well, I'll need that to add you to Drexel AI to contribute. If you have any questions, I'll be around for a little bit. Otherwise, I'll see you next week, if not sooner, in the Slack. Cool. Bye, everybody.
Bye. Thank you.
Bye, guys.
Yeah, you got it. I don't think. Do you have a question?
Yeah. Nope.
Nope. Okay.
I'll see you then.
I'll see you.
Also, are you free on 30th, 2 to 3 p.m.? Because I'm meeting Professor Kim then.
Why would you need me 2 to 3 p.m.?
I'm meeting Professor Kim, so I thought we could discuss some projects and stuff.
Oh, with him?
Yeah.
Yeah, for Drexel AI?
For Drexel AI and also for the GAN project. The review article, I also want to discuss that with him.
Okay. Yeah. I should be fine. That'll be right after work for me.
Yeah.
That's fine.
Yeah.
Cool. All right.
Cool.
I'll see you.
Okay. I'll see you then. Bye-bye.
Peace.
Have a nice night.
You too.